Good morning, everyone. Good morning, good afternoon, or good evening, whatever it may be, whichever time zone you are in. According to that, I'm saying. So, this topic is based on cytokines, like regulation of immune responses, immunology portion, and this particular study note is developed by EPG Patshala team, MHRD, Government of India. So, I would like to express my sincere gratitude to the development team for developing this study note. Thank you so much. So, let's learn about cytokines. After reading this module on cytokines, the students will have clarity on the various aspects of cytokine properties, their classification, structure of the various cytokine receptors, the similarity of cytokines with hormones and cytokines. Chemokines has also been discussed for further understanding. Since the action is highly specific at very low concentrations, it is imperative to comprehend the antagonists of cytokines to exploit their therapeutic potential. The biological actions of cytokines are tightly regulated and their impacts on Th subsets have also been discussed. So let's try it. Let's have an introduction about this topic. The most versatile defensive mechanism of organisms can be determined by the recognition of a variety of stimuli. The most versatile defensive mechanism of organisms can be determined by the recognition of a variety of stimuli monitored by suitable response in terms of initiation of immune response. The most versatile defensive mechanism of organisms can be determined by the recognition of a variety of stimuli monitored by suitable responses in terms of initiation of immune responses, both innate and active. The immune function is based on the biological activities of innumerable glycoprotein molecules called cytokines. Classically, cytokines display extensive functional activities that arbitrate both effector and regulatory immune functions, combined with far-reaching effects across an array of biological systems. Cytokines have an impact on both host defense and an array of normal physiological and metabolic processes. Cytokines are low molecular weight protein mediators that are produced transitorily post-cellular activation. Typically, their action is restricted between neighboring cells and spreads over a short range of humoral regulators. So, the most versatile defensive mechanism of organisms can be determined by the recognition of a variety of stimuli. The most versatile defensive mechanism of organisms can be determined by the recognition of a variety of stimuli monitored by suitable responses in terms of initiation of immune response, both innate and adaptive. The immune function is based on the biological activities of innumerable glycoprotein molecules called cytokines. Classically, cytokines display extensive functional activities that arbitrate both effector and regulatory immune functions, combined with far-reaching effects across an array of biological systems. Cytokines have an impact on both host defense and an array of normal physiological and metabolic processes. Cytokines are low molecular weight protein mediators that are produced transitorily post-cellular activation. Typically, their action is restricted between neighboring cells. Cytokines are low molecular weight protein mediators that are produced transitorily post-cellular activation. Typically, their action is restricted between neighboring cells and spreads over a short range of humoral regulators. Cytokines may modulate the functions of each cell, thereby regulating processes happening under normal and pathological conditions. These molecules were earlier called the tumor necrosis factor, TNFs, interferons, interleukins, and growth factors that are involved in almost all biological processes, from immunity to inflammation to cell proliferation, angiogenesis, migration, and repair. Historical perspective. The earliest information was reported in the mid 1940s as soluble factors released by white blood cells, primarily lymphokines, to differentiate from monokines in an endeavor to categorize the soluble factors from their primary sources. In the late 1970s, they were classified as cytokines with the introduction of molecular methods that ensured the cloning of IFNs. So, in the late 1970s, they were classified as cytokines with the introduction of molecular methods that ensure the cloning of IFNs. Carl Weizmann's group identified IFN alpha and David Goedel's colleagues, and IFN beta was initially isolated by Tada Taniguchi. I'm not sure about the pronunciation, barring the erythrocytes, each cell can produce and subsequently respond to a cytokine. By mid 1980s, numerous cytokines with their corresponding cytokine receptors that could be explicitly pre-mediated by using molecular tools like antibodies and cDNA probes. Only some bioactivities in cell supernatants were assessed simply by activity of macrophage activating factor, lymphocyte activating factor, and leukocyte pyrogen that was prevalent in the 1960s and 1970s. By the mid 1980s, the advancement in the tools enabled researchers to assess the expression of cytokines in pathological as well as physiological conditions. The term cytokine has evolved from the Greek kaitos, meaning hollow or vessel, and kainin means to move. That was initially used to differentiate a group of immunoregulatory proteins, interleukins, from other chemicals known as growth factors that modulated the cell proliferation and bioactivation of non-immune cells. They possibly regulate the intensity and duration of immune response by stimulating the activation, proliferation, and differentiation of cells. Regulation of secretion of antibodies and cytokines. The classical immunomodulatory cytokines are able to affect proliferation and differentiation in both immune and non-immune cells. So, typical growth factors are directly or indirectly able to regulate immune competent cells. There is a major group of soluble cytokines that are chemoattractant protein moieties called chemokines that include molecules such as monocyte chemotactant proteins or MCPs regulated on activation, normal T expressed and secreted, RANTES, and lymphotactin that are responsible for the activation of immunoinflammatory cells. Then cytokines as hormones of immune system. The term cytokine encompasses those cytokines secreted by lymphocytes known as lymphokines and monokines to those secreted by monocytes, macrophages. Although these other two terms continue to be used, they are misleading because secretion of lymphokines and monokines are not limited to lymphocytes and monocytes but encompass a variety of cells. Generally, cytokines are called interleukins. That refers to the ability of the secretion of leukocytes that may act on other leukocytes too. Interleukins 1 to 25 have been identified and the list is overgrowing. So some cytokines are known by trivial names, including the interferons and tumor necrosis factors. Recently, chemokines that play an important role in the inflammatory response has gained remarkable prominence. So cytokines share many properties with hormones and growth factors, and hence their properties are often blurred. So all three classes are secreted as soluble factors that elicit their biological effects at picomolar concentrations by binding to receptors on target cells. The definition of a cytokine as a soluble factor produced by one cell and acting on another cell in order to bring about a change in the function of the target cell was based on the endocrine system of hormones. Cytokine may be considered as a hormone of immune system, but the properties of cytokines are different. Hormones are generally the primary secretors of a particular cell or tissue, but cytokines are secreted by maximum number of types of cells. Remarkably, on a molar basis, cytokines are far more effective than hormones. The concentration of a cytokine IL-1 that induces gene expression and synthesis of cyclooxygenase 1 COX2 is 10 pm, and the ability of IL-12 to induce IF and gamma is 20 pm. Growth factors are produced constitutively, hence differ from cytokines and hormones as they are secreted in response to specific stimuli. The secretion is short-lived, generally ranging from a few hours to a few days, while hormones normally act at a long distance in the endocrine manner, but cytokines generally act over a short distance in an autocrine or paracrine manner. In addition, mostly hormones are secreted by specialized glands and have their exclusive action on one or few types of target cell. Alternatively, cytokines are generally produced by a variety of cells and possibly bind to a number of cells too. Properties of cytokines. Pleiotropy is mediated by a cytokine that has diverse effects on various target cells. When more than one cytokine exhibits similar functions, they are said to be redundant. That makes it challenging to attribute a specific activity to a single cytokine. That is redundancy. Pleiotropy is mediated by a cytokine that has diverse effects on different target cells. When more than one cytokine exhibits similar functions, they are said to be redundant, which makes it challenging to attribute a specific activity to a single cytokine. When the combined effect of two cytokines is more than the added effects of an individual cytokine on cellular activity, it is termed as synergism. So antagonism is exhibited by cytokines where there is inhibition of one cytokine or equalizes the influence of another cytokine. Alternatively, when the influence of an individual cytokine on a target cell induces that particular cell to produce one or more additional cytokines that may induce other target cells to produce supplementary cytokines, it causes cascade induction. Since these molecules and their respective receptors provide vital signals for important processes, any aberration in the cytokine or in the signaling pathways initiated by them or other receptors may be the cause of numerous diseases. The half-life of the secreted cytokine is generally very short, ensuring that they act for a limited period over short distances. They are not performed and stored, but are formed by new gene transcription per the cell's need. Pleiotropism activated CD4 plus helper T cell, IL4, B cell, IgE protection, CD4 T cell, Th2 differentiation, macrophage inhibition. Then redundancy, IL2, IL4, IL5, B cell proliferation. Synergy, IFN gamma, TNF, increased expression of class 1 MHC molecules on many cell types. Then antagonism, IFN alpha, IL10, macrophage activation, inhibition of macrophage activation, IL10. Cytokine exhibit pleiotropy, synergy, redundancy, antagonism, and cascade induction that ensure the regulation of cellular activity in a synchronized and coordinated manner. Next, classification of cytokines. Based on the structure, cytokines have been classified in 4 different families. Classification of cytokines. Based on the structure, cytokines have been classified in 4 different families: immunoglobulin family, hematopoietin family, interferon family, chemokine family, and tumor necrosis factor family. Cytokines are documented numerically in order of their discovery, like interleukin IL-1 to IL-35 by any particular functional activity, like granulocyte colony stimulating factor, tumor necrosis factor, TNF, by its role in inflammatory response, innate or adaptive, early or late, pro-inflammatory or anti-inflammatory, primarily as per the origin of cell, like monokine derived from monocyte, lymphokine derived from lymphocyte, and more exclusively and conclusively by the shared structural homologies with interrelated molecules. So here you could see the classification. Cytokine superfamilies display homology, share sequence similarity, and some resemblance in their respective receptor systems, but lack functional similarity. Cytokine superfamily also have significant cell membrane receptor ligand pairs having regulatory functions and showing evolutionary pressures that use shared structural motifs in various immune functions. The immunoregulatory cytokines are TNF, INF, TNF receptor superfamily 1, including lymphotoxins, cellular ligands like CD40 and TNF alpha mediate B cells and T cell activation, but FAS-L CD95 stimulate apoptosis. The interleukin IL-1 receptor superfamily 2 contains cytokines like IL-1 alpha, IL-1 beta, IL-18, IL-33, and IL receptor antagonists that arbitrate physiological and host defensive functions. This family also includes a series of mammalian pattern recognition molecules called the toll-like receptors that play a crucial role in early innate response for recognition of microbial species. Interleukin 1 was initially considered to induce fever-regulated lymphocytes, caused joint destruction, and damaged bone marrow cells. 2 distinct proteins, IL-1 and IL-2, were confirmed, and 10 ligands of IL-1 have been recognized, termed IL-1F1 to IL-1F10. All their genes are present on chromosome 2, bearing IFN4. IL-1 is important in both innate and adaptive immunity, and an essential effector of the host inflammatory response, especially in natural immunity. Activated mononuclear phagocyte is a major source of IL-1. Additionally, fibroblasts, dendritic cells, B cells, endothelial cells, astrocytes, epithelial cells, and large granular lymphocytes secrete IL-1. Bacterial and viral infections are the major causes of IL-1 induced fever as it relaxes endotoxins, TNF, IL-1 per se, or contact with CD4 plus cells further generate IL-1. It is responsible for the acute phase reaction in response to infections. Primarily, IL-1 destroys the beta cells leading to type 1 diabetes mellitus by inhibiting the function as well as promoting the apoptosis of B cells of pancreas. IL-2 secretion is induced by the activation of T helper cells, and IL-1 stimulates B cell activation. Enhanced IL-1 concentration has been observed in autoimmune diseases as IL-1 exert their effects via specific cell surface receptors that include a family of about 9 members characterized as IL-1R1 to IL-1R9 exhibiting an intracellular TLR domain except IL-1R2. The ligands for all of these receptors have not yet been identified. The next, cytokines that stimulate hematopoiesis. Cytokine produced during natural immunity and antigen-induced specific immune responses exert strong stimulatory effect on bone marrow stem cells. The maturation of hematopoietic cells is dependent on cytokines that are also responsible for the progressive division and differentiation of pluripotent stem cells. Different types of colony-stimulating factor CSF act at various stages of development of bone marrow cells and promote development of cells of different lineages. Some actions of CSFs are influenced by other cytokines such as leukotrienes, LT, tumor necrosis factor, TNF, tissue growth factor beta, TGF beta, and interferon gamma. Interleukin 3, IL-3. It is also known as multi-lineage colony stimulating factor, multi-CSF, that acts on immature bone marrow progenitor cells and promotes the expansion of all cell types. The receptor consists of a WSX-WS unit and a 150 kilodalton signal transducing unit shared with IL-5 and GMCS. IL-3 promotes the growth of mast cells derived from bone marrow progenitors. Granulocyte macrophage colony stimulating factor, GM-CSF. GM-CSF is a 22 kilodalton glycoprotein secreted by activated mononuclear phagocytes, fibroblasts, T cells, and vascular endothelial cells. Its receptor and the signal transducing unit are similar to the ones for IL-3. In humans, GM-CSF acts on non-committed form of leukocytes, red blood cells, platelet cells, and macrophages. As it is not detected in circulation, its action is mostly local, that is, bone marrow or at the site of inflammation by vascular endothelial cells, mononuclear phagocytes, and fibroblasts. Then monocyte macrophage colony stimulating factor, or MCSF, also referred to as CSF1, is secreted by endothelial cells, macrophages, and fibroblasts. The secreted polypeptide is a dimer of approximately 40 kilodalton. MCSF acts on progenitors that are already committed to develop into monocytes. Granulocyte colony stimulating factor, GCSF, activated T cells and mononuclear phagocytes, fibroblasts, and vascular endothelial cells secrete GCSF. The secreted polypeptide is 19 kilodalton and its receptor is made up of WXX-WS unit. It acts on progenitors of bone marrow which are mature and already committed to develop into granulocytes. It is found in circulation and brings neutrophil maturation and release from bone marrow during inflammatory reaction occurring outside the bone marrow. Then IL-7 or interleukin 7. Interleukin 7 is secreted by bone marrow stromal cells and acts on hematopoietic progenitors committed to the B lymphocyte lineage. It also stimulates the growth and maturation of immature CD4, CD8 T cell precursors in the thymus. T and B precursor cell maturation and survival are dependent on IL-7. It is also required for positive selection of CD8 plus thymocytes and regulation of T cell receptor gamma. While IL-7 is an effective stimulator of immature B cell expansion, but it is not enough to promote precursor T cell division. Cytokine receptors. Further signal transduction pathways are triggered when cytokines bind to precise receptors on the cell membrane of target cell that eventually alter gene expression in the target cells. The response of the target cell to a particular cytokine is assessed by the manifestation of definite membrane receptors. Th cells secrete cytokines that directly influence Tc cell and B cell activities. Sensitivity and high affinities of biological effects mediated by cytokines can be accessed at picomolar concentrations. Their dissociation constants range from 10 raised to minus 10 to 10 raised to minus 12 m, exhibiting their high affinity. They are located primarily in the vicinity of the secreting cell, and their action is commenced via specific receptors expressed on the cell membranes of their target cell. They are designated as juxtacrine, autocrine, and paracrine response modifiers. Cytokines differ from hormones as cytokines are produced by cells that are not structured as spatial glands and that act holistically to affect the entire biological phenomena such as wound healing, inflammation, oncogenesis, and organogenesis. Autocrine action, paracrine action to the nearby cell, endocrine action to the circulation, distant cell. Types of cytokine receptors. In an autocrine action, a particular cytokine binds to cell membrane receptors of the very cell that's secreted. Alternatively, it may bind to receptors on the target cell that lie in close proximity to the producer cell, thereby setting up paracrine action. Sometimes it may bind to target cell in other parts of the body, exerting an endocrine function or action. So, chemocytokines may regulate the duration and intensity of the immune response by stimulating or hindering the initiation, proliferation, and differentiation of numerous cells, and by modulating the secretion of antibodies or more cytokines. The binding of given cytokine elicits immune response from target cells either by enhancing the expression of cytokine receptors or by secretion of supplementary cytokines that may in turn affect other target cells too. Therefore, even a few antigens or cytokine activated lymphokines may further influence the activity of different cells involved in the immune response. For example, cytokines produced by activated Th cells can influence the activity of hematopoietic stem cells, Tc cells, macrophages, natural killer cells, B cells, and granulocytes, thus activating a gamut of networking cells. Cytokines have been categorized into different classes based on their structural analysis that include the trimeric tumor necrosis factor TNF family in helical cytokines, the cysteine node growth factor, and the beta trefoil growth factors. Cytokines may also be classified based on the type of receptors that they are able to bind. Based on the shared structure, the cytokine receptors may be categorized into 6 main families: class 1 cytokine receptors, class 2 cytokine receptors, TNF receptors, IL-1 receptors. Tyrosine class 1 cytokine receptor family has 3 shared receptors in the common gamma chain and the common beta chain, which precipitate in the formation of receptor complexes for nearly 20 different cytokines. Structural classification of cytokines and receptors: ID type, TNFR type, cytokine R type 1, cytokine R type 2, cytokine R type, chemokine R type. Structural classification of cytokine: IgG type, TNFR type, cytokine R type 1, cytokine R type 2, chemokine type R type 1. Interleukin type family. Cytokine families: interleukin type 2 family, interleukin type 1 family, TNFR, IgG family. Okay. Homology of shared cytokine receptors and their interaction. Significant information regarding the mechanism of action for cross-reactivity of the receptors shared by cytokines have evolved from studies of structural complexes between cytokines and extracellular domains of their respective receptors. GPI30, gamma C, and beta C express a conserved core structure for the assembly of the extracellular cytokine receptor signaling complexes. However, there are many significant aberrations among these systems, thereby resulting in considerably diverse signaling intricate topologies. For the assembly of extracellular cytokine receptor signaling complexes, however, there are many significant aberrations among these systems, whereby resulting in considerably diverse signaling intricate topologies. So, the class 1 cytokine receptors or hematopoietin receptors form the major assembly among the cytokine receptor family and are membrane proteins with an extracellular N-terminal and an intracellular C-terminal. The extracellular segments exhibit a flexible architecture and has the characteristic 200 residue long cysteine producing homology region having a conserved binding motif for cytokines as structurally defined In the human growth hormone receptor complex, two fibronectin type 3 domains interconnected by a linker are present in the CHR module and denote the typical recognition module for all helical cytokines that may be existed on all type 1 cytokine receptors. The upper N-terminal domain consists of 4 conserved cysteine residues forming the stranded disulfide bond kinase receptors and sometimes even chemokine receptors. The conserved TRP, SERX, TRP SER motif is present on the lower C-terminal domain. A few key cytokines such as interferon, IFN gamma and IL-5 have 2-4 helix bundles forming 8 helix architecture. The mere binding of cytokines induces oligomerization of the receptor, leading to the opposition of the intracellular domains of the signaling moieties. Unlike many growth factor receptors, insulin epidermal growth factor that possesses tyrosine kinase activity at the same intracellular domain, the receptors of class 1 cytokine do not exhibit any inherent enzymatic activity, but their intracellular domains are constitutively linked with tyrosine kinase of the Janus kinase family and to a more derived degree the TYK kinase. JAK, TYK kinase are advanced by ligand-induced receptor oligomerization that results in self-phosphorylation along the intracellular domains of the receptor. Once tyrosine residues are phosphorylated in the receptors, they further act as the docking sites for alternative family of proteins, the signal transducer and activator of transcription STAT proteins. Binding of STATs to the intracellular domains of the receptors causes the phosphorylation of tyrosine residues with subsequent detachment from the receptors. Dimerization of the phosphorylated STATs occur. This then translocates into the nucleus where they finally bind to DNA recognition sequence to act as transcription factors. Moreover, the major JAK STAT signaling pathway, the class 1 cytokine receptors also use other signaling transduction mechanisms such as PI3 kinase, RAS, RAF, MAP kinase pathway, and the insulin receptor substrate. The structural aspects of JAK STAT communication during receptor signaling lack clarity and represent a major future leading edge in receptor structure morphology of cytokines. The critical question is either case is their significance as therapeutic targets and the prodigious message is that anti-cytokine medicine is a rapidly growing field with major pharmaceutical impact. Cytokine antagonists. Biological activity of cytokines can be inhibited by a number of proteins that can possibly act in two ways, either enhancing the effect they bind directly to a cytokine receptor but fail to activate the cell, or downregulating it. They bind directly to the cytokine, inhibiting its activity. IL-1 receptor antagonist, IL-1RA, is the most characterized inhibitor is that binds to IL-1 receptor but has no activity. The antagonistic property is evident as binding of IL-1RA of the IL-1 receptor blocks binding of both IL-1 alpha and IL-1 beta. Therefore, production of IL-1RA may play an important role in regulating the extent of inflammatory response and has been exploited for treatment of chronic inflammatory diseases. Interestingly, chemokine inhibitors may be present in the extracellular fluid or even bloodstream. These are soluble antagonists that may possibly be the remnants of the extracellular domain of cytokine receptors created by their enzymatic cleavage. Production of IL-1RA may play an important role in regulating the extent of inflammatory response and has been exploited for treatment of chronic inflammatory disease. Interestingly, cytokine inhibitors may be present in the extracellular fluid or even bloodstream. These are soluble antagonists that may possibly be the remnants of the extracellular domain of cytokine receptors created by their enzymatic cleavage. Commonly isolated soluble cytokine receptors have been reported are those for interleukins IL-2, 4, 6, 7, interferon, IFN, gamma and alpha, TNF alpha and LIF. Leukemia inhibitory factor is an interleukin 6 class cytokine that affects cell growth by inhibiting indifferentiation. When LIF levels drop, the cells differentiate. The soluble IL-2 receptor SIL2R released in chronic T cell activation has been characterized. The alpha subunit releases a segment containing the amino terminal 192 amino acid when treated by proteolytic enzymes, resulting in a 45 kilodalton soluble IL-2 receptor. This shed receptor has the ability to bind IL-2, therefore preventing its interaction with the memory-bound IL-2 receptor. The presence of SIR2R has been used as a clinical marker of chronic T-cell activation and has been observed in a number of diseases such as autoimmunity, transplant rejection, and AIDS. Viruses may also produce cytokine-producing proteins or mimics of cytokines. For example, pox viruses has been reported to encode a soluble TNF binding protein and a soluble IL-1 binding protein. Both IL-1 and TNF have broad spectrum anti-inflammatory activity response. These soluble cytokine-producing proteins possibly reduce the inflammatory effects of the cytokines, thereby giving an advantage to the virus. Reports discuss that Epstein-Barr virus produces an IL-10-like molecule, viral IL-10 or VIL-10, that may bind to the IL-10 receptor, and like the cellular IL-10, suppress Th1 type cell-mediated response that are generally effective against various intracellular parasites. Such molecules produced by viruses that mimic cytokines help the virus to circumvent the immune response, favoring the survival of the pathogen that eventually help them to overcome the formidable barrier of host immunity. That's kind of dangerous. Regulation of cytokines. Cytokines are produced in the Golgi and probably transported via the endoplasmic reticulum to be processed into cytosolic forms and can transverse intracellularly, remain membrane bound, or released as soluble mediators in the surrounding milieu. So cytokines mediate autocrine functions either by reverting to the nucleus, where they may act as a membrane expression, transcriptional regulators, or direct ligation of the receptor on the secretory cell or intracellularly inside the secretory cell. The distance and kinetics of the effector function are restricted by various factors, including physiochemical parameters of the peptide per se, or the existence of soluble receptors, enzymatic degradation, or extracellular matrix binding, or unique cytokine binding proteins in inflammatory milieu. Then a role of cytokine in disease. Neuroinflammatory, neurodegenerative disease, Parkinson's, multiple sclerosis, respiratory asthma, chronic obstructive pulmonary disease, COPD, CVS, hypertrophy, ischemia, reperfusion injury, then GIT, IBD, IBS, ulcerative colitis, I am not sure the pronunciation, renal, glomerulonephritis, diabetic nephropathy, then liver, viral hepatitis, alcohol hepatitis, fever, IL-18, TNF, rheumatoid arthritis. Cytokine remedies. Interferon in treatment of viral disease cancer. Several cytokines used to enhance T cell activation in immunodeficiency disease. IL-2, IFN alpha, TFN alpha, IL-2 and lymphokine activating killer cells. LAC, anti-cytokines in management of autoimmune disease. Anti-TNF antibodies in treating septic shock. Anti-IL-2RA in treating adult T-cell leukemia. Anti-IL-4 is under trial for treatment for allergies. Cytokine secretion of Th subsets. The cytokines that are produced by Th subsets have very distinct functions. Th1 derived cytokines elicit cell-mediated immune functions, whereas Th2 derived cytokines promote humoral response. Cell-mediated, humoral. Okay. Cytokine secretion patterns determine the type of immune response to a particular antigen. Th1 and Th2 cell can be distinguished by the cytokines that they secrete. However, both the types secrete IL-3 and GM-CSF. Th1 secrete IFN gamma that activates macrophages. There is an increased microbial activity. IFN gamma also upregulates the level of class 2 MHC molecules that induces class switching to IgG that supports phagocytosis and complement fixation. The TNF beta and IFN gamma both mediate inflammation, while IL-2 and IFN gamma secreted by these cells promote differentiation of fully cytotoxic TC cells from CD8 plus precursors. Th2 secrete IL-4 and IL-5 that help in the production of IgE and supports attack mediated by eosinophils on helminth infections. IL-4 also helps in class switching to IgG that does not activate complement. The switch from IgM to IgE is mediated by IL-4. IL-4 is also needed for the development of Th2 response. The cytokines produced by Th1 and Th2 promote the growth of the subset that produces them and inhibit the development and activity of the other subset. Such cross-regulation accounts for the inverse relationship between antibody production and cell-mediated immunity. Th7 extracellular bacteria, fungi, autoimmunity. Th1 cell-mediated immunity and inflammation, intracellular pathogens, autoimmunity, inflammation. Th2 antibody-mediated immunity, extracellular parasites, asthma, allergy. Treg immune tolerance, lymphocyte homeostasis, regulation of immune response. Then cytokine cross-regulation. Two subsets Th1 and Th2 are regulated by the cytokines and exhibit two distinct effects on subset development. Either they enhance the growth of the subset that produces them or they inhibit the growth and activity of the opposite subset that is defined as cross-regulation. For instance, IFN gamma secreted by the Th1 subset particularly inhibit proliferation of the Th2 subset, and IL-4 and IL-10 secreted by the Th2 subset downregulate the secretion of IL-12, one of the critical cytokines for Th1 differentiation by both macrophages and dendritic cells. So correspondingly, the cytokines have opposite effects on target cells other than Th subset. IFN gamma secreted by the Th1 subset promote IgG2A production by B cells but inhibits IgG1 and IgE production. On the other hand, IL-1 secreted by the Th2 subset promotes production of IgG1 and IgG. IgE suppresses production of IgG2A. The spectacle of cross-regulation elucidates the belief that there may be an inverse relationship between antibody production and cell-mediated immunity. That is, when antibody production is high, cell-mediated immunity is low and vice versa. Now let's have a conclusion. It has almost been 7 decades since the first cytokine was pronounced. Ever since, more than 400 cytokines, growth factors, and chemokines have been described with innumerable functions regarding not only the immune system but on having an effect on every organ system of the body. However, despite this, our understanding how these factors alone, in conjunction with other factors, influence homeostasis and inflammatory events is rather new. So that area is open to research. So I would like to encourage all of the viewers of this video to research on this topic, and cytokine antagonists are also a very important topic for research. Hence, concerted research is therefore imperative in order to clarify understanding the complexity of these pathways and the pleiotropic interactions that symbolize cytokine signaling.